Hello everyone. So I'm Rutvi and I was recently a part of this program that was held by Pine Biotech named Bioinformatics for Infectious Diseases. This whole was a program which taught about certain parasites, viruses, bacteria like corona, Ebola, rhino, tuberculosis and malaria. It also explained us in depth about diseases like AIDS, malaria, flu, tuberculosis, guinea virus, etc. One of the main implications in this program was to bring in the application of bioinformatics in treatment, research, sequencing, and understanding the infectious disease and host pathogen interactions. This whole program was a session included into 10 parts. There were 10 sessions twice a week, and the mentors for the program were Ilya Brodsky, who is the co-founder and CEO, Dr. Mohit Mazumdar, who is a PhD in computational biology, and Dr. Harpreet Kaur, who is a PhD in bioinformatics. She was as our bioinformatics trainer. Dr. Mohit Mazumdar was partnerships and project mentor, and Ilya Brodsky was the one who led this all sessions. So, talking about the purpose of this program, the purpose of this program was generally to get into depth and understand about certain parasitic genomics, viral genomics, and how they have evolved over the past few years, their actions and their courses. Second, one of the most important purpose of the program was to investigate our immune responses, how we react to certain pathogens. Third was finding the biomarkers, like when any type of infectious disease is caused. Biomarkers indicate a very important tool for vaccine design and drug design. Fourth was to study the microbiome study, that is the structure, function of host and pathogen. And third was the course of infection, which included host pathogen interactions, how the host invaded our immune system, the artificial ways, the types, the phenomena, and further on. So the main program of it was the infectious disease program. So we basically taught the difference between infectious and non-infectious diseases, which, as we know, infectious disease is spread through contact, that is from person to person, and it is contagious, and it is spread by pathogenic microorganisms like virus, bacteria, parasites, fungi. Non-infectious diseases are non-contagious and they do not spread by contact. Further, they are spread by environmental factors, nutritional deficiencies, genetic inheritance, mutations, lifestyles, etc. There is a particular way by how pathogens spread. It might be by replication in their own breed or colonization in particular environment. It can be due to cell death, and there can be a chance of bacteria or virus entering into our immune system because certain types of immune cells are dead. It can also be due to organ function damage or the release of certain harmful toxins which could prevent the use of certain microbes into our body. This is the whole picture I have picturized. This is used to understand first how the pathogens interact and replicate. What techniques are used for the sequencing, the genome sequence, and the application of the genome sequencing. Now we know that NGS is a system that transforms the data sequences, the genome sequences into data file formats like FASTA, FASTQ, BAM, H1, etc. So further, this genomes of any pathogen is further used for getting a pure insight into virulence capacity of that particular parasite. It is also used for studying the protein expression, and using this protein expression, vaccine design and drug design is done. They're also used to study the population structure and evolutionary history of that particular parasite, which gives a comparative analysis of genome sequencing and strains of same species, if they are evolved or not, are more virulent or not. It is also used for studying specific sequences of strain detection. It is also used for diagnostic purposes of how they have used by PCR arrays or DNA microarrays, and proteins are immunoprecipitated using protein expression, which are further used for serological tests of vaccine development or development of 3D structures of protein, which is further used for drug design. Moving on, and for the session 3 and 4 and 5, we were given a hands-on tutorial about how to make a demo pipeline of MSA, that is multiple sequence alignment. Over here, we were also taught about how to use certain platforms like NCB virus viral genome platform, where we can download the genomic sequences and alignment trees of certain viruses, not certain but almost all viruses according to their year of virulence. Further, we were also taught some of the methods and concepts of replication. Also, we were taught methods of RNA sequence alignments, which were known as targeted amplification, which is the first one and involves a gene or a conservation of specific genome, which is very highly variable. Second was total DNA or RNA, that is by microarrays, which helps to know certain known genes. Third was by amplicon and environmental conservation, this method which is used to conserve hypervariable regions. So this was taught in third, fourth, fifth with an hands-on session on making a demo pipeline. So this is a picture which shows the mutation variant parallel analysis of mutation variant. So this is a demo pipeline we ran, and furthermore we downloaded the genomic sequences of certain viruses like Ebola, Plasmodium, Plasmodium falciparum, etc. And then we further made the mutation pipeline to find out what type of mutations are present in which gene, the lipid content, etc. There were many more pipelines we had run. This is just an example. In session 7, 8, and 9, and 10, we were taught about the host pathogen interactions. In host pathogen interaction, we understood the mechanism by which the microbes interact and invade our immune system. We also learned about RNA sequence overview, which included three main steps, that is pre-processing, mapping, and quantification of expression levels. Using this quantification of expression levels, we understood about certain type of protein function, that is how amino acid sequences, what is the 3D structure, what is the high throughput data. Now, amino acid sequencing had further branches. We taught about the sequence similarity, dissimilarity, and the features of the particular amino acid sequence. The 3D structure told more about global and local types of proteins and what proteins are used, what are the isoforms, etc. In high throughput data, we learned about protein interaction and gene expression on the same platform. Using differential expression and network analysis, we were further able to study the gene expression in depth of any particular type of organism, say it a human or any model organisms. So this was taught, and after this 10 sessions, we were given a 2 weeks time for which we have to think about the project we would like to work on bioinformatics of infectious diseases. The project title and hypothesis could be any, and our mentors told that they would be helping with taking this project further. So overall, it was a very good session and had a very deep insight on how infectious disease play an important role together in hand with bioinformatics. Thank you.